Okay, everyone. Welcome, everyone, to this week's Towers seminar. My name is Daniel Knopf. You likely don't see me. We had some issues and my camera is off. It's a pleasure to welcome Maria Zawadowicz. She is here from Brookhaven National Laboratory in the Department of Environmental and Climate Sciences, so a colleague from close by. And she joined the department in 2020. A few words to our speaker. She got her bachelor degrees in chemistry and physics with distinction in Lake Forest College in Illinois, and then went straight to PhD at MIT, and I believe with Dan Cisco's group, looking at ice nucleating particles, so quite related to my research interests. And she graduated in 2017 from MIT. That was followed by a postdoctoral research stint at Pacific Northwest National Laboratories, where we have also a lot of colleagues with whom we collaborate in Washington State. That was for 3 years, from 2017 to 2020. Maria has got some honors and awards during her studies. She has a famous Jules Jarny Prize. Jarny is an eminent person in our community, or was at MIT, got also the NASA Earth and Space Science Fellowship in 2014 to 2017. Maria, for her stage of her career, is very productive, has over 20 publications in the last 5 years. That is quite impressive. And so I'm very happy to have her here talking about aerosol particles, their chemical composition, measurements done at the atmospheric radiation measurement observatories, where some of us, including me, also perform measurements or use their data for, diagnostic analysis of models. So welcome, Maria, and the title of her talk today is Long-Term Characterization and Source Apportionment of Non-Refractory Submicrometer Aerosol Chemistry at DOE Atmospheric Radiation Measurement Observatories. Please, Maria, take control over my computer to show your presentation.
All right, thank you so much for the introduction, Daniel, and thank you all for coming. So as you now know, I normally work on the same island as you do, but currently I'm actually in a ski lodge at Crested Butte, Colorado, and it is not for skiing. We have an instrument here, and I'll actually talk more about this field project later. Okay, let me there you go. Oh gosh, yeah. So not to take away from this wonderful introduction, but just to reiterate, I'm Maria Zawadowicz. I'm an assistant environmental scientist at Brookhaven. I'm also an ARM instrument mentor, and I'll explain what that means in a little bit. And my interests are primarily mass spectrometry of atmospheric aerosols, including their hardware, the deployment in the field, as well as the science that we can do with these instruments, centering primarily on biosphere-atmosphere interactions. So in order to talk about the mass spectrometry of atmospheric aerosols, I will just cover briefly the question of what is an atmospheric aerosol. So probably most of you already know. So an aerosol is a suspension of solid or liquid particles in air. An alternative name for the atmospheric phenomenon of aerosols is atmospheric particulate matter, so PM1, PM2.5, usually used in the context of air pollution. And in this electron micrograph, I really like showing it because it just shows a wide variety of atmospheric aerosols. Their size, with the very small soot particles, and larger, I think these are organic or sulfate particles, which are these larger blobs on the order of a micron, to their morphology. As you can see, the Soot particles are really intricate network of smaller particles. And in the C panel, they're actually perfectly round fly ash particles. And this complicated figure from Woodby and Cantrell shows this pretty much also the variability in aerosol size and composition in a different way. Aerosols span several orders of magnitude in size. The smallest sizes are usually associated with formation of from vapors through condensation. And then coagulation in this middle size mode. And then for your large particles, we usually see windblown dust, sea spray, and other particles that are emitted from the environment. So one reason to study aerosols is that they have an effect on climate. And this figure is from the previous IPCC report, the AR5. And the relevant thing that I'd like to point out here is that for aerosol radiation interactions, and especially for aerosol cloud interactions, the error bars are really high, meaning that these aerosols, understanding aerosols and their interactions with radiation, water vapor, and climate limits our ability to model the Earth system and therefore predict future climate. Now, as you know, recently the AR6 came out, and the error bars specific for aerosol effective radiative forcing are no less small. So we are still grappling with this problem. So aerosol effects on climate can be generally sorted into direct and indirect categories. For direct aerosol effect, it is the interaction of aerosol particles directly with radiation. So in this example, for sulfate particles have have a large real index of refraction, which causes them to be reflective. They reflect solar radiation, contributing net negative radiative forcing. But for example, a black carbon particle will be absorptive. It has a higher complex index of refraction. And therefore it's a positive radiative forcing. And in this particular example, they cancel each other out. But so aerosols can interact with solar radiation in both ways. But this is a pretty straightforward problem. We generally know how to model this since we know the refractive indices of these particles. What's more complicated is that aerosol particles also interact with water vapor and therefore making clouds. They can either make cloud droplets or ice crystals. And the composition of the cloud, is it a liquid or ice cloud, will determine how it's going to interact with the radiation, as well as its altitude and a myriad other factors. This is the indirect effect, and it is generally much less well known than the direct effect and contributes more uncertainty. So its classic example of indirect effect is the Twomey effect, or the first indirect effect, where introducing more aerosol particles into the environment produces clouds with more smaller droplets, which increases the cloud albedo. So you can think about natural aerosol abundance perturbed by something like human activity, introducing a change in how the clouds interact with radiation. And this is a dramatic demonstration of this effect. These are ship tracks. So ships introduce a lot of small soot particles into the marine boundary layer, thus perturbing the natural abundance of the aerosols and making these clouds. And now, as you can see, those ship tracks are much more reflective than the background cloud around them. Okay, so these are just generally what aerosols do to climate. I will now talk a little bit about the mass spectrometry of aerosols. So in this micrograph again, this particle, which is sulfate or organic, is on the order of 10 picograms of material, that it's very small. So we're dealing with very small particles, a very small mass of actual analyte. And we need a technique with a high sensitivity to those chemical components. Another thing to know about aerosols is that they are not static. Once an aerosol is emitted into the atmosphere or once it forms in the atmosphere, in the case of secondary particles, it will continuously change. For example, in this cartoon, a particle which could have been, say, dust, is interacting with atmospheric vapors such as isoprene or alpha-pinene and oxidants such as ozone to develop an organic coating. So we need a technique with a fast response and in situ capabilities to really capture those transformations, as opposed to, for example, a filter technique, which will capture the coated particle, but may not give us an insight that this was when the coating formed. So mass spectrometry, so in situ mass spectrometry of aerosols generally satisfies both of these requirements. We have high sensitivity to analyte, and they are in situ, the instruments, at least the ones that I will be talking about. So in general, in situ mass spectrometry of aerosols needs several different components. We need an inlet to introduce aerosols into the high vacuum where they are processed. And generally, the sample inlet separates the aerosols from the gas that carries it inside the instrument. Inside the instrument, we ionize the particles, or more accurately, we usually vaporize and ionize them in either 2 steps or 1 step. And we need ions because the next step is that we accelerate the ions with a high electric field. And once accelerated, they enter a drift region, which can have a field or not, depending on how the mass spectrometer is built. And here, larger ions will move slower than lighter ions. And based on their kinetic energy, we can detect the mass to charge ratio and therefore separate the different aerosol components based on their mass. And therefore based on their chemistry. And here is a cartoon which does a better job of explaining it. Okay. So for example, the instrument that I work with the most these days is an AMS, is an ACSM actually, which is a variation of an instrument called AMS or aerosol mass spectrometer, built by Aerodyne about 20 years ago now. And as you can see, it does have the inlet. It's an aerodynamic lens. It subsequently sends particles into this red element at the end, which is a heated surface at 600 degrees C, which vaporizes them. There's also a filament that emits electrons, which ionizes them by electron impact, therefore making ions, which are then analyzed by the mass analyzer. In this case, it's a quadrupole, but we also have TOF instruments. And this particular instrument has a very long deployment history. So this figure is already several years old and it's very out of date, but those instruments are in situ capable. So they have been deployed around the world, generating data like this. So the pie charts represent non-refractory aerosol composition. So either organic, sulfate, ammonium, and nitrate in different places. So what I'd like to talk about a bit right now before I move on to my current research is how exactly do we go from this complicated piece of instrumentation under high vacuum to a pie chart like this. It requires a lot of abstraction and a lot of understanding of what a mass spectrum is and how it relates to chemical compositions. So when we deploy an aerosol mass spectrometer or a different type of mass spectrometer, we collect aerosol particles and we produce a mass spectrum like this. It's essentially a graph of intensity on the detector versus a mass to charge ratio. And by analyzing the spectrum, we subsequently go to a pie chart like this of, for example, the non-refractory aerosol composition. But there are multiple steps in between to actually get this result and incorporate it into an atmospheric climate model. We have to take into account that every instrument has uncertainties. Every measurement has additional uncertainties beyond the instrumentation specific ones. We have to consider whether the experiment was designed correctly. And we have to impose a correct classification of abstraction level onto these mass spectra. To make this clearer, I'll give now an example of something that I did during my PhD. So this is not an AMS actually. It's an instrument, which is also an aerosol mass spectrometer, which instead of a heated surface and electron impact uses a powerful UV laser to ionize particles, therefore accessing certain chemical components that are not accessible by AMS, such as dust, like silicates or soot. And when we sample bioaerosols with this mass spectrometer, this is what results. It's a mass spectrum. It has many different fragments. Most importantly, it has some phosphorus fragments, which turn out to be the key to recognizing bioaerosols in the environment. So in order to take mass spectra taken in the field and classify them as bioaerosol or non-bioaerosol, we developed a database of many different mass spectra using both biological and non-biological materials. So biological materials could include things like bacteria or fungal spores or pollen. And then non-biological materials included things like dust, which naturally contains phosphorus to separate it. And we were able to design a machine learning algorithm to separate biological and non-biological mass spectra based on certain marker combinations. And this can now be applied to a large environmental data set. So this is a series of flights that NOAA did over the past 10 years or so. The oldest campaign is from 2009. And we analyzed all this data. Actually, the oldest one is from 2004. We analyzed all this data to derive vertical profiles of bioaerosol, which ended up agreeing with the model fairly well. And they were also fairly extensive measurements of bioaerosol in the upper atmosphere specifically. Okay, so, and this example is mostly to drive home the point that once you obtain a mass spectrum, it's generally not the end of it. It has a lot of, there's a lot of science in just accessing, in just interpreting the mass spectra that come out of these instruments. So now I'll move on to my current research. I currently work at BNL, and about half my job is spent as an instrument mentor for the atmospheric radiation measurement user facility. It's a DOE user facility in a similar way that a particle accelerator may be a DOE user facility, but this facility is not, it's movable. It is essentially a collection of atmospheric observatories that are either fixed or move around the world to provide high quality atmospheric data sets. Specifically, I work with the group that deploys the aerosol observing systems, or AOSs. Here are some photos and schematics from a paper that my colleague Janek wrote in 2019. And the specific AOS pictured is the one at the Southern Great Plains site. And I will come back to this AOS. An AOS is essentially a large shipping container with an inlet stack to sample the aerosols. And it also has air conditioning and various instruments deployed inside. So I will specifically focus on the ACSMs, but there are other instruments deployed within an AOS. As an instrument mentor, I take care of our mass spectrometers. Currently, there are 3 quadruple ACSMs. 2 are deployed at permanent ARM observatories at Southern Great Plains in Oklahoma and Eastern North Atlantic in the Azores. There's one mobile AOS that has a quadruple ACSM. It is currently deployed in La Porte, Texas, as a part of the Tracer campaign. There are also 2 time-of-flight ACSMs, both assigned to mobile observatories, one currently in Colorado, where I am as well, and one will be going into a new southeastern United States site that you may have heard about. There's also one PTR TOF, which breaks the mold of these instruments a little bit because it's actually a gas phase instrument. And it allows us to measure trace gases such as isoprene, which are aerosol precursors. As an aerosol mentor, I also take care, I curate the ACSM datasets that are currently available. And the currently available ACSM datasets that I've been doing quality control on are from these 4 deployments. So SGP and ENA are the permanent ones. Cacti and LASIK are recent field campaigns. And here are the Jimenez style pie charts of these particular places. So green is organic, red is sulfate, yellow is ammonium, and blue is nitrate. Oh, and pink is chloride. Soon I should be able to also release a data set from one of the longer term deployments at Olectok Point in Alaska as well. We also have 2 ongoing field deployments. One of these is SAIL in Colorado and Tracer in Texas. The SAIL deployment stands for Surface Atmosphere Integrated Field Laboratory. Its PI is Daniel Feldman, and it's a Colorado River Basin study. So I'm here in Crested Butte to do some of the remaining installation on the AOS that will be here for the next 1.5 years. And the other ongoing field deployment is Tracer, so tracking aerosol convection interactions experiment. A PI is my colleague from BNL, Mike Jensen, and the AOS in La Porte, Texas is currently operational. And the ACSM specifically has been collecting data for a month. And some of this data is actually already available. So if you are specifically interested in Houston atmosphere, and which is very interesting because it sits at that, because it's an interaction between very polluted city atmosphere, as well as some marine influence from the Gulf. Come and talk to me and we could start looking at the data. All right, so right now I'll talk a little bit about the data set acquired at SGP, because it is something that I've been working on for a while. So at the Southern Great Plains Observatory is one of the fixed ARM sites in northern Oklahoma. And these are some photographs of the site. As you can see, the site is very rural. It sits among fields, like agricultural fields. There is not much other activity there. So generally, the atmosphere is pretty clean, apart from the agricultural activities. So here's a pretty busy slide of all the data that we have. So the ACSM at SGP has been now deployed for about 10 years. And apart from some data gaps, it actually recorded a pretty impressive record. So this is a data set that I've been working with the most. It was deployed at SGP in late 2010, and it remains working to this day. We have just recently refurbished the instrument and brought it back into operation. So it's currently also collecting data. Here are some closures that I did between the ACSM and co-located sensors within the AOS. It sort of spent 2 decades of AOS operation when the AOS was almost completely rebuilt. So for example, the SMPS to which I'm comparing in the third panel, it was a different SMPS at the beginning in red, and a different SMPS currently in blue. And you may notice that there are actually some differences between the two. It is most useful if we have chemical data at the site to compare it with the ACSM. High-scale campaign in 2016 actually brought an AMS to the site. So in these upper right 2 panels, you see a comparison between the ACSM and the AMS and the SMPS, showing that at least in 2016, the ACSM actually agrees with the AMS really closely. And that is what we really like to see, as those measurements are pretty difficult to make and they don't always agree. What I'm comparing here, by the way, is aerosol volume, which makes it easy to compare to SMPS volume distributions. But for AMS specifically, we can actually compare the mass loadings from ACSM and AMS, and those also agree pretty well. During 2019 and 2020, some improved filters were also collected at the SGP site. And both a volume closure and the chemistry closure between the improve and ACSM also works really well. So this is sort of a spot checking of the instrument performance, but it does appear that at least from 2016 on, the ACSM agrees with other sensors pretty well. We can also do a bit more involved comparisons. So for example, here I derived kappa from the ACSM measurements as I take the chemical composition in terms of organic sulfate, ammonium, and nitrate, and derive kappa using a volume balance and known kappas for inorganics and organics. Using the ACSM derived kappa, I then develop a closure between the CCN measured and the CCN predicted based on the critical diameter calculated from the ACSM derived kappa and integrating the SMPS size distribution. And those generally work pretty well across all the supersaturations that this CCN is measuring. It's a ramp style measurement that circles through 6 different supersaturations. And it generally shows pretty good agreement. And here's a bit more about the SGP ACSM. So just looking at this species fraction graph, where I colored it by the composition, you can see that there is a very strong seasonal trend in the SGP data. Generally, during the summer, we have a lot of organic, which is likely SOA formation, both at the site and transported. It's accompanied with also an increase in sulfate. But in the winter, we have a strong increase of in inorganics, specifically nitrate. And that's a pretty interesting effect that I haven't seen elsewhere. And previously, it's been explained as an actual agricultural influence, so from ammonia, ammonium nitrate. But I don't have a very clear explanation for why it shows such a very strong seasonal cycle. And this is already interesting, but we can also do a bit more on the analysis of ACSM organic spectra specifically. So each data point in these two graphs, time series graphs, is a mass spectrum, much like this one. So each one of these points is actually a vector of many different measurements at each mass to charge ratio, 150 in this case. And that's a very big data set. And we can actually extract patterns from this using an approach called positive matrix factorization, which has been used in our field to analyze this type of data for a while. Please ignore this triangle. I thought I deleted it, but I'm schematically showing here X as the data set, which contains a time series of many different mass spectra where each row is a time point and each column is a mass to charge ratio. So going across, these are mass spectra collected at different time points. And technically, we should be able to decompose such a matrix into two matrices, G and F, in which one of them describes time series, and then the other one, F, describes profiles. So we are extracting profiles, characteristic profile, source profiles from the matrix and describing their time series. And you may notice that this is really non-negative matrix factorization, but the positive matrix factorization differs from some of the more widely used methods based on what quantity exactly is minimized during this calculation. So in PMF, we minimize the quantity Q, described by in which the error matrix is rescaled by the measurement uncertainty sigma in this case. Now, for a data set this big, we can't just take the matrix and decompose it because this is a very big matrix. So the trick that we play is that we perform PMF in small windows. So there is a rolling window applied to the data set. We translate the window. So the window is 14 days long, so 2 weeks, and we translate it a day at a time. So there's some overlap between the windows, which is something that we want because overlaps or that helps us derive the uncertainty in this decomposition. And what I'm actually using here is a bit more complicated than just one pass PMF because I'm adopting a method from a recent paper by Lina Heikkinen et al., where we do rolling PMF on this large data set once. We take results from each window and cluster them using standard clustering techniques, which derives characteristic profiles, which we can then put back into the rolling PMF as constraining factors to arrive at the final OA model. So PMF can be initialized from random values, but it can also be initialized from already known profiles. And when such profiles are known, it's called a chemical mass balance. In this case, it's the right approach because in analysis of atmospheric aerosol, we actually have a wealth of knowledge about how these profiles look and what they correspond to. And in picking the profiles, we can leverage some of the previous work that has been done on the topic. So here is a fairly preliminary result of PMF of SGP dataset, the all 10 years of it. There are some gaps in this PMF because in some places, in some early days of the ACSM operation, the data quality wasn't as good as later. But generally, past 2016, it is a very robust decomposition. Here we identified 4 different source profiles, one of them corresponding to biomass burning, second corresponding to hydrocarbon-like OA. Biomass burning is pretty self-explanatory. It comes from combustion. Hydrocarbon OA usually comes from things like traffic. It can also come from combustion, however. And in this case, the two are very correlated, so they are likely from the same source. We also have semi-volatile OA, which is SVOA, which in this case is very likely to be fresh SOA formation. And we arrived at this primarily by comparing this factor to an AMS factor derived during the high scale campaign, which is very similar. And because the high scale also had a PTRMS, they actually named this an isoprene OA, as likely this fresh SOA is formed from isoprene. We can't say that with certainty from this data set, as we don't have isoprene measurements over this whole period, but this likely corresponds to a fresh SOA. And then finally, the low volatility OA is very oxidized organic aerosol, which could come from any of these sources once they're oxidized enough. So once the aerosol has been in the atmosphere for a long time, ozone and OH oxidize it to pretty much look the same. And this is a low volatility OA. One of the checks on this analysis is examining the cycles, yearly cycles of these profiles. So biomass burning generally has a peak in the spring and summer, where burning would be expected. And in the case of SGP, this may be agricultural burning from the nearby fields, especially if it occurs in the spring. Very similar profiles for hydrocarbon OA, which suggests that these are pretty much the same source. And then for semi-volatile OA, there is a distinct peak in the summer, which is expected for SOA formation. For low volatility OA, it's a combination of the previous two. There is a peak in the summer, which is a transported SOA perhaps, and a peak in the spring, which could be transported biomass burning. So from once these PMF decomposition is done, we can use this data to explore some of the atmospheric regimes and trends at SGP. And this is what I'm currently doing with the data. So for example, based on the biomass burning time series, we can clearly delineate periods, polluted periods when there is high BBOA and clean periods when there is low BBOA, which corresponds to the background at SGP. And in this case, we're asking whether or not these are different by looking at the fraction activated from CCN. So ratio of CCN number concentration to overall number concentration from the SMPS. And we see a little bit of an effect with BBOA at high supersaturations, but it's not super strong. If we ask the same question about SVOA or the fresh SOA, the effect is actually stronger. So starting at 0.4 supersaturation, we see that during the periods with high SVOA, we also see more particles activated. It is not, which is very preliminary, but could suggest that SOA formation drives the CCN budget at the SGP site. Okay, so this is pretty much what I'm doing right now. I'm playing around with the SGP data. I welcome any interaction. If you are interested in any of these topics, please don't hesitate to reach out. There is definitely a lot of data, and I'm really looking forward to delving deeper into other ACSM datasets as well. I also have other projects that are currently underway. I am pretty soon we'll have a single particle mass spectrometer in the lab as well. And I'm looking into getting back into bioaerosol work as well. So as a ACSM mentor, I will be remiss in not telling you about the fact that most of these datasets that I talked about, with the exception of the PMF, are actually available on the ARM website. Here's a quick screenshot and a link. And if you're interested, please look at the data. If you're unclear, if you are unsure of what the flags mean, or if it can be used for a project that you have in mind, please reach out. And this is all I have. Thank you.
Thank you, Maria, for this nice talk and the introduction in this quite complicated instrumentation. And I have to say, I mean, running these instruments, mass spectrometer instruments, even in the lab can be challenging. And then running them for years in the field consistently, producing this nice data set is much more challenging. So thank you for that. The floor is open. I will try to look at, I have now control back on participants, and if you maybe raise your hands, then I will shout out to you, and you can, with your name, and then you can just ask your question. That's maybe quickest. Do I see anyone? Ah, yeah, Paul Shepson, of course. Please go ahead.
Hi, Maria. That was great. I enjoyed your talk. Thank you. I'm interested in your observation of peak in nitrate aerosol in the wintertime. Your PMF didn't show any wintertime peaks. I'm wondering if it might be N2O5 chemistry, that maybe in the wintertime you have poorly mixed boundary layers and NOx sources around, like from flares. I don't know if there are flares in that environment, but sort of looks like it might be N2O5 chemistry. Have you looked at that?
Yeah, so the PMF was only run on the organic aerosol. So it does suggest inorganic chemistry. And yeah, I think N2O5 is a very good suggestion. I'm really unsure of what exact chemistry is behind these peaks, but because they're so clear, I think I'd like to sort of dig more into it. N2O5.
Is there NOx data from that campaign?
Unfortunately not. No, there are no NOx. There's no NOx. Yeah.
Okay. Anyway, very interesting. Thank you.
Anyone else? Do I see? I don't see a hand right now. Ah, yes, Alison, please go ahead.
Yeah, that was an excellent talk, Maria. It's really great to see that very long time series of SGP data very neatly laid out. I think the closures you've done to spot check the consistency among the data is, that's really great to see as well. I know that you focus mostly on the chemistry, but I'm wondering if you have plans for relating the chemistry to the long-term optical property data sets that we have.
Yeah, I would love to do this. I'm currently, so I've looked a little bit at the nephelometer time series, but I would need some of the, actually, we should talk about it as well. I'll need some help in exactly what assumptions to make to relate the two. But yes, that could potentially, it could actually potentially extend this data set even further, since optical properties have been measured at the SGP site for many years now.
Yeah, well, this is a very valuable database you've put together. Thanks.
All right. If no hand is up, maybe I ask a question. So I really enjoyed also seeing this long-term data set. And I had a question to your last slide where you analyzed the CCN active fraction in the data. And you said something at the end, it's maybe driven by the SOA. Sorry that my camera is not working anymore. As you took control over my laptop, somehow my camera switched off. So just bear with me. But you said it's driven by the SOA. And in all your data sets, I was astonished even other sites, sulfate is always present, it seems. And there's more sulfate in summer. And so I'm wondering, sulfate is of course very hygroscopic. And if you said the CCN activity is driven by the SOA at SGP, but if there's still sulfate present in the aerosols, wouldn't that dominate the overall hygroscopicity? And, or what kind of assumptions go in to derive or do this closure? Now, of course, and then my follow-up, maybe more a comment, because maybe that's not evident from the data. Of course, mixing state makes a big issue then if the sulfate is inside, and you alluded to that in your introduction, and the organic is outside and the water doesn't see the sulfate, then you would model hygroscopicity needed for cloud formation very differently.
So it is a big shortcoming of this instrument that it doesn't see the mixing state. So this could be sulfate coated with organic, or it could be sulfate and organic in different size modes. Currently, we can't tell. And it is a good point that an increase in SOA also corresponds to an increase in sulfate, and therefore this could be a completely coincidental correlation. But yeah, unfortunately, mixing state is difficult to tell with this instrument, but we can perhaps do some simulations to see how the mixing state would impact this analysis.
Yeah. Therefore, it was more a comment than a question. One would need complementary instrumentation maybe to nail that down. I think, Paul, your hand is up again, or is it still up? Then you have the floor.
Sorry, that's a legacy hand.
Okay, all right. Okay, no, thank you, Maria. I just wanted to put this thought out. Anyone else? Okay, it looks not right now. Then I would thank Maria once more for this great talk. And